Okay, the first question, who coined the term mitosis? Fleming. DNA synthesis takes place in S phase. When does pairing occur in chromosomes? Zygote. Next, most active stage? Interphase. Which of the following is a method of cytokinesis in the plant cell? A and C, phlegmoplast or cell plate. What happens in interkinesis? Interkinesis ki asile? Interkinesis is after the completion of meiosis 1 and before the beginning of meiosis 2, the phase of resting is called as interkinesis. What happens? Preparation for the second meiotic stage. What is very important about interkinesis? It is a phase where DNA replication does not take place. Because if you talk about I phase or interphase, that is a phase when DNA replication takes place, especially in the S phase. Interphase boli koa loge loge tar logot DNA replication associated. Into interkinesis koa loge loge tar DNA replication nohoy. Right? Next, division of nucleus is indirect in. Division of nucleus is indirect in. Indirect actually কিহৰ কাৰণে কৈছে কাৰণ cytokinesis প্ৰথমতে karyokinesis তাৰপিছত cytokinesis mitosis yeah huh question number 7 নকল নেকি 6 নে 7 oh 6 I have just skipped in which stage of the cell cycle nucleus becomes bigger? S. When will the nucleus become bigger? When the nuclear contents increases. Division of next is number 7. What happens in interkinesis? Preparation for the second meiotic division. Number 8, division of nucleus is indirect in mitosis. Who coined the term? It is secondary MAC. What can be observed in interphase nucleus under the light microscope? Okay, interphase nucleus or ki? You can see the genetic material, right? Genetic material sabo lagile ami ki koribo lagbo? You have to stain them out. Right? If you stain, there are certain parts which will take more stain and there are certain parts which will take less stain. So, first আমি কোনটো দেখা পাম যোনটো darkly stained, so it is heterochromatin. Hetero kyo? Chromatin divided into how many types? Euchromatin, heterochromatin. Heterochromatin kiho karone dekham কাৰণ they are darkly stained. Right? So, june basically wrong to lobo microscope or hetu agote bhalke dekha pam no. Euchromatin part to lightly stained. So, iman clearly to visible no hobo. Right? Who divided cell cycle in 4 stages? It's Howard and Pell. How many parts of cell cycle formed by interphase? 90%. Minimum number of meiotic division need to produce 64 pollen grains. 60? Ki likhisa? No, it should be 64, no? এইটো question কেনেকৈ আমি কৰিম 1 microspore is equal to 4 pollen grains, right? Then 16 into 4. Number 14. 13 কি হ'ব তুমি কোৱা এটা সেলে 4টা ফর্ম কৰে তো 16 টাই কেৰা ফর্ম কৰিব সেইটোৱেতো কৈছো 64 হ'ব লাগে এটা ভুলকে লিখা আছে ঠিক আছে ওকে হ্যাঁ it is 16 in cell cycle changes of which stage are not visible interphase which type of division leads to polyploidy endomitosis which of the following does not occur in anaphase 1 but occurs in anaphase 2 15th question endomitosis polyploidy mane ki jeta tar chromosomes tar dna content bilak bari bari goi thakibo right dna content baribo kintu bostu to separate hoi na jay so as a result the ploidy keeps on increasing right jeta n twice n thrice n 4n no haploid diploid polyploid so polyploid condition ketia hobo one condition is endomitosis mo agote koisu no nuclear division hoy kintu within the nuclear membrane right ekane separate hoi na jay he karone ki hobo the number of genetic material will keep on increasing number 16 what happens in anaphase 1 but does not happen in anaphase 2 splitting of centromere anaphase 1 or splitting hoy no hoy Anaphase 1 or splitting hoy na hoy? Splitting hoy na separation hoy? Separation of the chromosomes and splitting of the centromere. If the centromere splits, what is the product? The chromatids separate, no? Or if the chromosomes separate, then homologous chromosome or duta part hoy jay, right? In angiosperms, endometriosis occurs in tapetum because tapetum cells are polyploid in nature. Crossing over takes place, pectitin. A contractile mid-body forms during cytokinesis in case of. Okay, this is a question related with the cytokinesis part. Constriction kuntu cellot hobo? Cytokinesis or cell wall constriction hoy no? Constriction kuntu cellot hobo? Plant cell or animal cell? Plant cell to constrict no kore no? That cell plate hai form kore. So in case of animal cell, the answer would be it divides into two by the process of constriction or Faro method, whatever you say. Next is question number 20. Which chromosomes may lose during cell division? Acentric chromosome. Why? Because centromere is absent. In which order cytokinesis occurs in plants? Centrifugal. Mazot prothomote tapsot sidole jay. Mitosis not occurs in shoot tip. Sorry, meiosis. Which of the two events restored on? Meiosis and fertilization. Number 23. 24. Indirect nuclear division boli kole mitosis and meiosis. Number of meiosis required to produce 100 ovules. 100 ovules dise 100 grains dise ki dise? Ovules dise. Jedi ovules diye tetihale okol to female part to hai. Grains dile hai duitar combination hoi pelai grain ba seed form kore, right? So yate ovules dise. So ovules hole ki hobo? 1 megaspore mother cell produces kiman megaspores? 1 megaspore mother cell divides and produces how many megaspores? 4. 4 functional jano. Eta hai functional. Tamane we can say 1 megaspore is equal to 1 megaspore mother cell is equal to 1 megaspore. So kiman ovules lagibo? Jiman ase kiman, right? So answer would be 100. Which cell lacks nucleus? Sieve cell. Which elements helps in spindle formation? Mn. Colchicine, a mitotic poison, arrest the cell division in metaphase. Arrest cell division mane? Centromere separate by splitting of centromere by chromosome separation ebilakok hobole nidiye. That is what is called as arresting. So because if you apply colchicine, what will happen? Polyploid condition. No? Chromosomes bilak bari goi thakibo. They will not get separated. Amitosis is a characteristic of lower organisms. Reason of chromosomal movement of anaphase, kinetochore and spindle fibers. I mean, who is responsible to move the chromosomes from the middle to the pole? Yate khudise soa anaphase ot ki hobo? Metaphase ot gute chromosomes bilak equatorial plane ot log lagi jay. After metaphase ki phase hai? Anaphase ot ki hobo? Chromosomes will be or the centromere will split or the homologous chromosome will separate in case of mitosis or in case of meiosis I am talking about, right? So tate he movement mane from the middle to the poles kune help korise? Spindle fibers help korise. Spindle fiber bro kot lagise? Kinetochore lagise. So who helps it is kinetochore and the spindle fibers. Nuclear envelope reappears in telophase because telophase is the opposite of prophase. Slipping of chiasmata, terminalization occurs in which stage? Dikinesis. No, is called. The process is called terminalization. Where does it occur? In dikinesis. Right? Duplication of chromosome without the division of the nucleus, endomitosis again. Which does not occur in prophase? Hydration of chromatin. Karon ki hoy? Dehydration hoy. Next 35. RNA and protein synthesis takes place in which phase of interphase? G1 and G2 phase. In which stage of cell division? Number of chromosomes best counted? Metaphase. Right? Metaphase is the time when all the chromosomes will get arranged in the equatorial plane and then you'll be able to count them all. Right? So that's the time. Next. Cellular structure was disappeared during mitosis, nuclear membrane and nucleolus. 38. Blue-green algae, bacteria. Meiosis takes place in reproductive cell. Replication of DNA, S phase. A cell is bound to divide if it entered. Obviously S phase. S phase opera khuri jabo nware, no? How many chromosomes shall be present in a diploid cell and meiotic anaphase if the egg cell has 10 chromosomes? Eto kora. How many chromosomes shall be present in a diploid cell at mitotic anaphase if its egg cell has 10 chromosomes? 20 hobo. Ne 10 hobo, ne 30 hobo, ne 40 hobo. See. Eta cellot kiman thakibo? 40. Both sides, na? 20 in one side and the 20 in other side, so that will make 40. Because the information has been given about what? Information has been given about the haploid cell. Ama calculation ki koribol dise diploid cell. So egg cellot kiman dise chromosomes? 10. Ne kiman? 10 dise. So if n equals to 10, twice n is equal to 20. Right? If 20 hoga, then mitosis or kotha koise ne meiosis or kotha koise. Mitosis 20 chromosomes or centromere split hobo. So 40 hoye jabo na jay. 20. Karon mitosis boli kole, the point that you have to stress is the separation of the centromere, right? And if the centromere splits, so a chromosome eta centromere ase, duta chromatid lagi ase, no? Jetia leke centromere log lagi thake ami duita part of ki boli kom chromatid boli kom. Once the centromere splits and the chromatids move to the two part, we don't use the word chromatid, instead we refer them to as chromosomes itself. So eta chromosome split kori pele actually ki form kore? Two chromatids, but what we say? Two chromosomes. Once they move to the two poles. Is the point clear? So very clear ne? Meiosis which occur at the time of spore formation. Excellent. Oh yeah, 44 mitosis. Bouquet stage, leptotin, synaptonymal complex, all the above. Phragmoplast. Phragmoplast, what is the role of phragmoplast? To form the cell plate during cytokinesis in case of a plant cell, right? At the end of anaphase. 48. At anaphase 2 of meiosis, its chromosomes contains one DNA already separate hoi jabo no? Separate no hobo jano anaphase. Anaphase separate 2 DNA to ki hole convert hoi jabo? 1 DNA? Anaphase 2, right? Anaphase 1 hole to hetu e thakibo 2 DNA hoi thakile hete. Yate ki hudise anaphase 2, anaphase 1 hoi gol 4n opra arambha hoise, 4n opra 2n hoise, 2n opra lastot ki hoise 1n. DNA to half hoi japo na jay, reduced hoi japo na jay, reduced hoy no. Suppose starting cell to ami 4n hisabe dhoriso. 4n to prothom daughter cell gita jetia form koribo tahantor ki hobo twice n, right? Tahate to akko divide koribo meiosis 2. So twice n to কি হলে হৈ যাব n. So ultimately চাৰিটা যোনটো সেল ফৰ্ম কৰিব তাহাঁতৰ কি থাকিব n. n to থাকিবই লাগিব কাৰণ মিওসিছত কি ফৰ্ম কৰে জার্ম সেলসে ফৰ্ম কৰে তো জার্ম সেলসটো হ্যাপ্লয়েড হ'বই লাগিব কাৰণ দুটা জার্ম সেল যেতিয়া ফিউজ কৰিব তেতিয়াহে টুৱাইস n ফৰ্ম কৰিব সো জার্ম সেল প্ৰডিউসিং প্ৰসেস কি বুলি কম আমি মিওসিস সো তাতে যোনটো সেল ফৰ্ম কৰে তাত ডিএনএ হ্যাপ্লয়েড ওকে নেক্সট kinetin or cytokinin duration of interphase ketaman questions okoman gk oriented interzonal fibers occurs in prophase to nohoy right early metaphase no hoy late prophase no hoy thakil ki anaphase interzonal fibers ki asile first you have to know the meaning then only you'll give the answer right ene to aru inky pinky ponky no hoy no you have the options and inky pinky ponky father head donkey to no hoy no so interzonal fibers ki hoy interzonal fibers are those fibers or proteinaceous structures that joins the two chromatids if the centromere splits then okoman tanile tat ki dekha pabo right so actually if you are talking more precisely if you want to know about interzonal fibers it is something that is seen in case of anaphase 1 anaphase 2 dekha na pao no anaphase 1 or duta homologous chromosomes thake no duta homologous chromosomes log lagi thakibo lage so tat ami ki dekha pam fibers kisman attach jun mane ihote kam ki koribo attachment right increases the rate of mitosis by reducing the duration of kinetin. What is kinetin? It's a hormone, no? We have talked about CK, G18 and kinetin, right? So kinetin dele ki hobo? Cytokinin er kam ki? Cell division fast kore. Cell division fast kore mane kuntu process slow kori dibo. When we talk about cell division, we have to talk about cell cycle also. Entire thing is a cell cycle. Cell cycle has two phases, the interphase and the dividing phase. If you want the cell to divide faster, something needs to get reduced, the interphase gets reduced. নহলে normally eta cell divide koribo lagile maximum homoy kot lage interphase ot lage no oh hetu etu tumak divide kori dibo no kinetin yate ki koise if you want to apply it from outside right next okay interzonal fibers or concept to chromosome separate korile interzonal dekhaba ne centromere separate korile interzonal dekhaba he bostu janibo laibo karon anaphase 1 ot ki hobo chromosome separate anaphase 2 t chromatid separate so interzonal ami ketia dekhapam jetia chromatid separate so anaphase 2 t ami dekhapam centromere to split korile duta part hoi jabo na jay he duta part or majot threads bilak jenake pizza khale cheese pao no so that's the thing next is question number kiman asile 51 during cell division microtubules because microtubules or what we are talking about is spindle fibers what is spindle fiber nothing but microtubules okay 52 see meiosis hole obviously crossing over hole exchange hobo dikinesis 53 huh
centromere do not divide during anaphase 1 tate to mane chromosome pura
centromere do not divide during anaphase 1 question ki hudise not true he hudise what is the question which of the statement is true ne which of the statement is not true read the question properly okay karon exam to there is a lot of chance to make mistakes true ne not true question to porhote underline kori loba so that you don't get confused understood no now
yes
Next is 54. 54 is GK oriented. Answer is A. Okay. Early morning. Early morning 9 to 10. Huh? Tetrad formation in meiosis occurs in. Tetrad ki asile? 2 dyad is equal to 1 tetrad, right? So where was it formed? In case of Pectitin. In plants, meiosis can be observed in sporangia. In anaphase 1, each chromosome composed of 2 chromatids. Gap between division phase and start of DNA replication is called G1 phase. Next, 59. Division of centromere occurs in anaphase 2. In meiosis, nuclear membrane and nucleolus disappear during Dikinesis is the last stage of prophase, right? Cell cycle remain arrested at cell cycle remains arrested. What is the meaning? Kuntu number 61. 61. M phase নহয়. M phase to divide কৰিব. Arrested মানে কি? Divide নকৰে. ঠিক আছে? So নকৰে কেতিয়া? G1. G1 ত নকৰিলে কি হ'ব? It goes back to G0. Right? So G0 is a part of G1 itself. So if a cell wants to divide, it will go to GS and then it go to G2. If the cell does not want to divide, from the G1, it goes and collects itself in the G0 phase. So whether the cell will divide or not divide, decision to ক'ত হ'ব? G1 phase অতে হৈ যাব. Right? Because G0 phase is a part of G1 phase. Mitotic poisons, all of the above. Spindle fibers which extend from pole to kinetochores are chromosomal or tactile fibers. The longest phase in meiotic 1 or meiosis prophase 1. Separation of homologous chromosomes, disjunction. Notun word paisa no? Disjunction to remember. The process is called as disjunction. During division, spindle fiber attached to which part of the chromosome? Chromomere কি আছিল? Chromosomes ত এই dotted structure বোৰ কিছুমান thread হেতু হে এটা ক্ৰোমোজোমৰ ভিতৰত আছে বস্তুটো ন কিন্তু স্পিন্ডল ফাইবাৰটো কোট লাগিব হেতুৱে খুদিছে সেন্ট্ৰোমেৰত লাগে তো সেন্ট্ৰোমেৰ কি হয় প্ৰাইমাৰী কনস্ট্ৰাকশন সো আনসার উড বি প্ৰাইমাৰী কনস্ট্ৰাকশন next is 67 2 in tetrad the number of non crossover chromatids is normally 2 tetrad কেটা থাকিব চাৰিটা পাৰ্ট থাকিব right চাৰিটা part if suppose we are talking about this as a tetrad দুইটাক আমি কি বুলি কম tetrad এটা কি বুলি কম dyad right so এটুৰ লগত ক্ৰসিং হ'ব এটুৰ লগত থাকিব বাকি কি থাকিলে এটু আৰু এটু বাকি থাকিলে নাই so কেটা বাকি থাকিলে দুটা Synaptonymal complex is a characteristic of paired meiotic chromosomes. Paired meiotic chromosomes মানে homologous chromosomes. Persistent nuclear membrane and internuclear spindle are the characteristics of. এইটো বস্তু ইমান নালাগে কিন্তু তথাপি cryptomitosis Karyoplasmic index this already B. Volume of the nucleus and volume of the cytoplasm indicates karyoplasmic index. What happens? Karyoplasmic index or karone ki hoy? Eta cell divide kore. Cryptomitosis pisot di more details. Nalage. Question number 71. Karyoplasmic index decreases. 72, synthesis of proteins, all the above. Different proteins get synthesized in different phases of interphase. 73, telophase. 74, the changes of karyokinesis in mitosis occurs in cytoplasm. The changes of karyokinesis है दिसे karyokinesis तो nucleus होय no Okay, question number 75. Question number 75. Question to ki dise aur baki khini swa. D. 4 and 4. 20. ভুল আছে 75 ইনফরমেশন কি দিয়া আছে বাইভ্যালেন্টৰ কথা রাইট বাইভ্যালেন্টত কি থাকে দুটা ক্ৰোমোজোম একেলগে থাকে দ্যাট ইজ বাইভ্যালেন্ট কেটা বাইভ্যালেন্ট আছে আঠটা বাইভ্যালেন্ট কি কৰিব ডিভাইড কৰি সেপাৰেট কৰি পেলাই কি হ'ব দুটা পোলছত যাব কেটা যাব আঠটাই থাকিব না থাকে বাইভ্যালেন্ট কেটা আছিল আঠটা ইয়াৰ পিছত এনাফেজ হ'ব ৰাইট এটা তো মেটাফেজত হৈছে ন এনাফেজ হ'ব এনাফেজত আঠটা ইপিনে যাব এটো কোনটো এনাফেজ 1 ৰাইট নেক্সট এনাফেজ 1 ৰ পিছত বাকি প্ৰসেছৰ পিছত সেল এটাত কেটা ক্ৰোমোজোম আছে এনাফেজ হ'ব এনাফেজৰ পিছত টিলোফেজৰ পিছত সেল ফৰ্ম কৰোতে ক্ৰোমোজোমছ কেটা আছে আঠটা এটোৰ পিছত কেটা সেল ফৰ্ম কৰিব দুটা সেল প্ৰত্যেকটো সেলত কেটা থাকিব আঠটা ক্ৰোমোজোম দিস ইজ আ পাৰ্ট অফ এনাফেজ 1 এণ্ড মিয়োসিস 1 দেন কাম টু মিয়োসিস 2 মিয়োসিস 2 এটা সেলত আঠটা আছে ৰাইট ইয়াতে কি আছে ক্ৰোমোজোমছ they will again split so ultimately 8 হ'ব নহয় বুজি পাইছা নে সেল তো আঠটা আছে ন আঠটা সেলৰ পৰা আঠটা suppose we are talking about no 1 2 3 4 5 6 7 8 right anaphase 2 কি হ'ব centromere to split কৰিব নকৰে তো দুটা পাৰ্ট হৈ যাব না যায় সো ইয়াতে actually কেটা আছিল আঠটা দুটা পাৰ্ট হলে আঠটা ওপৰত আঠটা তলত হ'ব নহয় So it is 8 and 8 hoine no hoy. Read the question again and see. Information that is given is about a bivalent. Who forms a bivalent? A homologous chromosome forms a bivalent. If you are talking about homologous chromosomes means what? One from the father, one from the mother. So yete chromosomes keta ase duta, right? Dutar eta pair. Huh? ইকুৱেশ্যনেল ডিভিজন হয়তো মিয়োসিস 1 ৰ পিছত তুমি যোনটো দুটা সেল পালা সেই সেলটোৰ ক্ৰোমোজোম কিমান আছে আঠটা যদি ইকুৱেশ্যনেল ডিভিজন হয় এই সেলটোৱে যেতিয়া ডিভাইড কৰিব সেই সেলটোৰ ক্ৰোমোজোম কিমান থাকিব বুজি পাইছানে নাই সো মিয়োসিস 2 এখিনি লকেটো মিয়োসিস 1 হৈ গল মিয়োসিস 1 ৰ পিছত সেল কেটা প্ৰডিউস কৰিলে দুটা তাহাতৰ ক্ৰোমোজোমছ কিমান আছে আঠটা A2 cell will undergo meiosis 2 this cell will also undergo meiosis 2 if this cell undergoes meiosis 2 as you have said equational division so the daughter cells should also have the same number of chromosomes so if it started from 8 daughter cells kiman thakibo 8 itself meiosis 1 ot chromosomes or number 2 separate hole right next is meiosis 2 chromosome eta separate hole meiosis 1 ot homologous pair to separate hole right meiosis 2 eta chromosome দুভাগ হৈ গল বুজি পাইছা centromere to split কৰিব ন meiosis 2 में हाँ वो हाफ हो जाएगा हाँ हाँ राइट अंडरस्टूड ने did not understand बुझिला no so answer কি হ'ব 8 and 8 next number 76 does not occur in division of centromere. Number 77 division of nucleus twice but replication of DNA only once. 78 after meiosis 1 the two chromatids of a chromosome are genetically different. Why they are genetically different? Because crossing over ho gaya, right? Ribonuclease prophase. Number 80 denomitosis dinoflagellates no mesokaryotes stain for cell division acetocarmine gap between meiosis 1 and 2 is called interkinesis. In animals cytokinesis is centripetal. Rate of cell division increases by application of cytokinin because that is the hormone, that main function of that hormone is cell division. Next question number 85 chiasmata appears during diplotin. Meiosis can take place in meiosis diploid cell. Sorry. What happens in synthesis phase during cell cycle? DNA synthesis. Number 88 reappearance of the nuclear membrane. Telophase because telophase is the opposite of prophase. What happens in crossing over? Exchange. Huh? Kunto. Linkage and crossing chromosomes নহয় exchange of genetic material exchange বুলি কব হা number 90 the in mitosis the spindle is bipolar from both the poles. Synthesis and storage of ATP molecule G2 phase. Both the phases can finally G2. Number 92 okay 92 D the cell cycle consists of a short interphase and long M phase. Short interphase to কেতিয়াও নহয় no 93 G1 phase histone protein S phase. The sequence of cell cycle G1S G2M. During cell cycle DNA replicates how many times? Once. The synthesis of spindle proteins occur during G2 phase. Spindle proteins যোনবিলাক protein spindle fiber form কৰিবৰ কাৰণে use হ'ব right এইবিলাক ক'ত form কৰিব G2 phase out হা centriole পৰা spindle fiber বোৰ উলাব লাগিলেও তো কোনোবা এটা protein লাগিব ন এই protein টো ক'ত phase বা interphase তে form কৰে S phase of centriole to duplicate কৰে হা next is question number 99 98 if meiotic division is restricted in G1 phase of cell the condition G0 M phase 100 DNA polymerase is active obviously DNA replication of সময়ত so S phase exit from cell cycle yeah 101A 102 Onion root tips. 103. Mitosis in animal cell is amphiestral. Amphiestral mane? Duta centriole opera duta ester. So we are talking about amphiestral, both sides. Ester formation on both the poles is called as amphiestral. Mitosis. 104. Contains the same amount of DNA and the same set of chromosomes as those of the parent cell. Ultimately to ekei hobo no. $106D. 100. Oh. When nuclear division takes place without cytoplasmic division, it results in a synocyte. Synocyte means multinucleate condition. When nuclear division takes place without cytoplasmic division, all nucleus to divide kori, number of nucleus increase hoye ase. Kintu nuclear division of pisat ki hobo lagisil? Cytokinesis hobo lagisil, kintu hua nai. Na, polyploid na hobo. Yate ami bostuto synocyte condition buli kom. Right? Synocyte kyo karon cytoplasm divide kora nai no? Right? Synocyte. Oh, fungi বহুত বস্তু আছে অকল ফাঞ্জাই নহয় ফাঞ্জাইটো আমি পঢ়িছিলো সিনোসাইটিক কন্ডিশন right 106 is D 107 recombination nodules okay pectitin recombination nodules pectitin minimum number of meiotic division এটা তো পারিম 125 how many generations are required এটাও করাইছো 7 To produce 102 pollen grains, how many meiotic divisions are required? 26. Karon, it will come in dot. 102 divided by 4. 111. 80. DNA duplicate koribo so 80 ho jabo picogram. Phragmoplast. Centrifugal 112, 113 spireme stage, early prophase. Eta ami discussion korim. Next, chiasmata are formed as a result of exchange. Structure of nuclear membrane helps in nucleocytoplasmic exchange. I think that is the last question or what? 142 114 essential parts of paired homologous chromosomes chiasmata mane that X no chiasmata কাৰ ভিতৰত হয় ক্ৰসিং ওভাৰ কৰোতে এটো হৈ যায় ক্ৰসিং ওভাৰ হৈ যোৱাৰ পিছত যেটা তাহলে সেপাৰেট কৰিব আৰম্ভ কৰিব তেতিয়া X স্ট্ৰাকচাৰটো আমি দেখা পাও কাৰণ পটককেটো সেপাৰেট কৰি না যায় no so X টো দেখা পাও that is called as chiasmata so কাৰ মাজত হ'ব পেয়াৰডৰ মাজতে হ'ব 112 Phragmoplast is formed by Golgi complex and forms dash to form a cell plate. Phragmoplast, what is a phragmoplast that will help in forming the cell plate, right? When we talk about the cell plate, we are talking about a plant cell. Plant cell does not show that constriction or the groove method, no? So all the vesicles will come and arrange. First, where will it get arranged? In the center. From the center, it will go towards the periphery. So that is centrifugal. So cell plate formation is always centrifugal in nature. Structure of the nuclear membrane helps in. What is the work of a nuclear membrane? Exchange. Nucleocytoplasmic exchange. Nucleus of cytoplasm. That's called as nucleocytoplasmic exchange. I think that's all, right? So we have completed the assignment discussion. Thank you.